Hey everyone, this is Dylan here, and today I'm going to go over blood cultures. I think this is the most important culture that we do in the lab. What do you think? So blood culture consists of two bottles, an anaerobic bottle and an aerobic bottle, and that consists of the set. And normally you draw two sets of blood 15 minutes apart from two different sites. So in total, we'll have 4 bottles in the lab that we incubate. And the way you incubate it in the lab is normally you have a machine, and you pop it on the machine, and that'll read it every certain amount of time, let's say 5-10 minutes or so. And it's reading a sensor on the bottom of the bottle, which is originally green, and it changes from green to yellow if it's positive via CO2 concentration. I think that's what changes the color of the sensor. So then once that is read positive, or if it is read positive by the machine, it'll give off an alarm, and then it's up to you as the medical laboratory scientist to work it up. And the way it gets worked up is you're going to do a gram stain, and you also inoculate it to a couple plates and then incubate them. We do like a blood and a chocolate plate. If it's aerobic, and then if it's anaerobic, you add your anaerobic plate, which would be your CDC. And so when you do your gram stain, you just read that, and normally you'll see something like gram-positive cocci in clusters. You can also see gram-positive cocci in pairs or chains. You can also see gram-positive coccobaccili, or your gram-negatives would be mainly gram-negative rod. That's the main one you get. And you can also see gram-negative cocci in clusters, which would be your Veillonella, which would be in your anaerobic bottle exclusively. So you would then report that gram stain morphology to the patient's nurse, and then antibiotics would be initiated empirically based on what you reported. And then normally also another thing we have is potentially an ID and AST system. So you can get an ID of the organism within 2 hours. So you can use something called the accelerate, which you can see here. And then that'll also give an antimicrobial sensitivity or susceptibility for the organism within like six hours after the ID. And then the treatment can be specialized for what the bug is susceptible to. So I think that's the most important test that we do in the lab, the blood culture. And as a medical laboratory scientist working it up, it's something that needs to be done quickly. And if you're ever unsure about the gram stain, just do it again, and maybe ask someone else to see what they think. If you over or under decolorized it, for example, because resulting that gram stain morphology is very important for what treatment the patient goes on immediately before you get an ID and then susceptibility. So thanks for watching. Hope you know more about blood cultures, and see you next time.